Good. The next speaker is Professor Aoneng Cao from Shanghai University in Shanghai.
Thank you. And this laser? Okay. Okay. Thank you. My name is Aoneng Cao. So this is C pronounced as in Dutch. So it's Cao. I'm from Shanghai University. So today I'm going to talk about our research. Actually, it's many years' work, only published recently. So the title is good nanoparticle-based artificial antibody created by conformational engineering. We call it Goldbody. I will come to this later, but I just want to point out this unique of this Goldbody comes from conformation engineering, which I will explain later. So we have see a lot of particles for nanomedicine applications. Usually you have to functionalize these particles. For example, you can label it with different fluorescent probes, or you can load drugs, or you can link with targeting groups, even antibodies. Yesterday, Professor Dampier has mentioned that this link may have some problems, especially for proteins and antibodies. If you do not link it properly, the function may lose. But on the other hand, because this nanoparticle is a big platform, you can link many the same function groups on the single particles. So this way you can increase the function very often by so-called multivalency effect. For those who are not familiar with multivalency effect, actually this picture can tell you everything about it. So multivalence effect works even at the large scale, but the problem is that multivalency does not create new functions either. So a fundamental question we have been asking for many years is that can we create new functions on nanoparticle with non-functional groups? So this has been done for billion years as the protein did. So in other words, can we make nanoparticles protein-like? So to do this, we have to learn how the protein works. Here shows an example of a very simple antibody-antigen complex. So the interface is perfectly matched. So that's why the protein function is very specific. If we look only those backbones, we can see that there is actually called CDR loop here, which is responsible for the binding of this antibody to the antigen. So actually, for many proteins, their function usually depends on a few key residues. You can regard all other amino acids as a framework. Their major function is to keep these function groups at the right position. So then we ask ourselves, can we use a nanoparticle as this kind of scaffold here, and we reconstruct the conformation of this action fragment. This way we may create artificial antibody or artificial protein. So this task actually function nanoparticle with peptide has done for many years. So for gold particles, it's very easy to realize it. You just add one cysteine amino acids at any position of the peptide. you will be very efficient link to the gold nanoparticles through the so-called gold-sulfur bond. But the problem is that most of the peptide is very flexible. So from an energetic point of view, the conformation space of this peptide is huge. So this is why even the active conformation is enthalpically favorite. But due to entropic penalty, most of the peptide, if you cut it from the natural proteins, is inactive. So how to control the conformation? After many years, we come up with a very simple idea. Yeah, we think if we doubling this peptide on the surface, then we can greatly reduce the conformation space. And if what's more, we have to control the span of the peptide here. So what we're trying to do is to use, for example, the peptide density on the surface to adjust the span. So if we do it correctly, we may find the optimal density to get the optimal or the best conformation, best span and the best conformation. Hopefully, we can reach and we can generate this kind of artificial proteins. So it's a very simple idea, but it took us several years to realize for the first example here. We just showed this a small antibody antigen complex. What we try to do is here, we graph this CDR loop on its golden nanoparticles. This is what we try to realize. Here is the design of the peptide. This is the peptide on the original antibody. So we add 2 cysteine residues at both terminals. Then we can efficiently link to this particle. As control, we have only 1 cysteine control and also a shuffle sequence control. So because of the time limit, I will quickly. So and this antigen is a lysozyme. So it's an enzyme. We can use if we do it correctly, we can completely inhibit the activity of this enzyme. So this one sign of the successful of our design. And as I mentioned before, we try to use density to control the conformation. So as shown here, indeed, we can find the optimal density for the best inhibition of this lysozyme. So this optimal density corresponding to the right conformation and the highest inhibition. We also use the SPR to determine the binding affinity between this antibody with the lysozyme, and we got a similar result. So the optimal density has the strongest binding here. Actually, we can this binding is very specific. If we label different immobilize different proteins on SPR chips, so we can only use Goldbody or binding to the target antigen, and there's only very weak non-specific binding to other proteins. And all the controls show only weak binding to all those proteins. And then the binding affinity can be calculated as sub-nanomolars, which is about 100-fold stronger than the original antibody we mimicked. So, of course, there's also some contribution from multivalence effect and the avidity effect. So, and the other problem is, another question we try to determine is the binding site for this antibody. Fortunately, there's a small inhibitor for this protein binding to the same cleft of this enzyme. So if the binding is on the same side, they should compete for this binding side. And we observed the competition here. So if we use this small inhibitor to occupy this position, then we can suppress the binding of this Goldbody to the antigen. And this means that this antibody really binding to the designed position and successfully reconstructing the original activity on this. So next we try to ask, can we use this approach to create some other antibodies to target other antigens? So the second example, we select a more vulnerable target. Many of you may be familiar, EGFR, which is overexpressed in many tumor cells and has been a target for anti-tumor treatment. This is the outer membrane part of this EGFR, and the dominant 3 is the position in the side for the EGF binding side. So this side is also the target side for the antibody development. Here we found a small antibody binding to this domain. So this is what we try to do. We graph this loop onto this particle. So and as expected, we also observed a density effect to control the conformation and the binding binding affinity to the immobilized antigen. And the binding is also very specific, as shown before. So this is our anti-EGFR Goldbody. So it's a specific binding to EGFR and not to other control proteins. And the binding is even stronger than the previous one. The determined affinity is to the 10 to the minus 11, which is about 4 orders of magnitude stronger than the original antibody. And it's also 2 magnitudes stronger than current FDA-approved and set maximum, which this is in the clinical use for cancer treatment. So as the first example, then this the binding side for the comparison. If our design is successful, they should compete with EGFR. And indeed, we observed the competition as shown here. And we also observe the binding of our antibody to the cell membrane, as shown here. Maybe it's not very clear. Then we used confocal microscope to show the overlap of the membrane EGFR and our antibody, and the overlap is very well. So it's also binding to the cells. So and the most important thing is it also functions well. As shown here, our antibody here is our antibody does not induce cell proliferation, but it can significantly enhance the EGF-induced cell proliferation. This means they may have some anti-cancer activity or applications. I want to mention that currently the antibody, the therapeutic antibody is actually the most successful drugs in the market, especially for anti-cancer treatment. But antibody has their intrinsic shortcomings. One is that most antibody has to be humanized for human application. But our Goldbody only use the binding small, very short binding part of this antigen. So there's no homogenization problem. The other shortcoming of natural antibodies, their stability is very poor. So they have to be stored at low temperature. But for our Goldbody, it's very stable. You even can boil it at 100 degrees and still maintain their binding capacity. So to summarize, I hope I convinced you that we demonstrate it's possible to create protein-like function as in the protein-like way. We call this conformation engineering. Basic conformation engineering is controlling the rotation of bonds, which is contrary to common bonding formation approaches. We believe this kind of approach, even just a start, is a very small step. It's open up great opportunities. And last, I want to thank my collaborators and my students, and I thank you all for your attention.